2 lectures this week and then a test next week, right? On that? Okay. And have a great lab today. We're going to meet over in our labs today, right? Yes. I can get back in swing of things for some. Okay. My lab days confused. So, we're going to meet down in the neuroscience lab. That's where we're having lab today, so 228 down the hall. And if you can bring a laptop, we're going to be entering data into a group data sheet during lab. Or I also have computers in there that we can pull it up. It's just when everybody's sharing those computers, it just takes a little longer. But we can do that as well. So either way. Anything else about it? No, that's it. All right, so the rest of this week we're going to finish talking about gustatory system. And, one other thing before we get on with this first. Your labs. So, I did take some time off during spring break. I did not work on the regular labs all of spring break. So, forgive me for that. I got about half of them done. I'll get the rest of them done this week and get them back to you. But what I thought about is, this is an upper level lab course. You guys have already learned how to do APA, hopefully, and you're demonstrating that and writing these lab reports. So, what I want to do is any section, so abstract, introduction, method, results, discussion, any of those sections that you get an A on, you don't have to write those anymore for the rest. You've demonstrated, I know how to write a good introduction, I know how to write a good method, right? I don't need to make you write these things over and over again if you can do it at A quality, right? If you don't get an A on that, then on the taste lab, say you got an A on your abstract and your introduction and methods, but not on the results and discussion. So for the taste lab, you just have to write the results and discussion, right? If you get an A on everything, then you'll just have to do a summary of what the lab was, what we found in the lab, right? So motivation to, if you didn't get an A on the section this time, to do well. And like I said, I'll try to get those back to you as soon as I can this week, probably Thursday or so. I'll get back to you this week. Cool? Makes sense? All right. Sounds good. So, the taste system, gustatory system, we left off last time, went through all of the sort of bottom-up signaling, got to talk a little bit about conditioned taste aversions, and today we're going to focus on the top-down influences on our behavior. Top-down influences on our behavior. And we'll start back with conditioned taste aversions. So we'll talk about aversions, cravings, and then more complex influences on that. So, just to remind you, as we mentioned last time, a conditioned taste aversion happens when a negative event, typically getting sick, is paired with having eaten some food item or drink item, right? And the more times that happens, we can have one trial learning, right? But if it happens multiple times, then that obviously becomes even stronger, right? And I mentioned that lesion studies showed that the PDN and the amygdala were necessary for conditioned taste aversion. So if you go in and ablate the amygdala, rats can no longer form taste aversions. If you go in and just ablate the PDN, then same thing. So you need to have both of those structures intact. They're important things. I also mentioned that you can use conditioned taste aversions as a research tool in 2 ways. One, if we want to know if you can taste the substance, right? If we want to measure, for instance, a rat's threshold. What is a rat's threshold to detect sucrose? Right? Something that they like. So I give a rat sucrose that I know that they can taste, make them sick, they form a taste aversion, and then the next day I can give them an array of sucrose solutions, some very diluted, some very low, and measure, okay, which ones will they lick, which ones will they avoid. They're only going to avoid the taste, right? So if they can't taste it, they won't avoid it. And on Thursday, I'm going to talk about the research that supports a role for fat taste. And in that research, we use conditioned taste aversions as a tool in order to show that rats could detect and avoid the molecules that are found in dietary fat. Right? So one way that we can use that as a research tool. Another way that we can use this as a research tool is to see if something makes an animal sick. Right? If we want to know, hey, does exposing an animal to stimulus X cause them to feel sick? If I give them sucrose and then expose them to that thing, if they then avoid sucrose the next day, I can say, hey, that thing made them form a taste aversion, right? It was a negative event in a way that got associated with that taste. And we'll talk about this when we talk about the vestibular system, and we'll talk about how we use this tool to identify whether being in a high magnetic field makes somebody nauseous, feel nauseous. In fact, this was used way back in the 50s or 60s when we were first trying to figure out if X-rays were aversive, and it turns out that you can expose monkeys to X-rays and pair them with, this is when we were first going into space, and so there's a lot more radiation in space, and so people were interested, hey, is this a negative event? And it turns out that monkeys will form taste aversions if they're exposed to high amounts of X-rays. So anyway, different ways that we can harness this learning in order to understand certain things. All right, so the ascending taste pathway that we talked about, we have 3 nerves, right, coming in from our oral cavity. All of those converge in the NST. And from the NST, that signal goes up to the PDN. This is in a rat brain here, not a human brain, obviously. Then through the thalamus, right, the ventral postmedial thalamus, up into the insular cortex for taste, and then up into the orbital frontal cortex, or prefrontal cortex for flavor perception. There's also descending pathways that are coming back down from areas like the nucleus accumbens and the amygdala. Right? So the PDN also, right, has this, so here's our taste flavor pathway. We also have a motivation and emotion pathway. It's coming in and going to places like hunger and satiety in the lateral hypothalamus, going to areas of reward like the ventral tegmental area and nucleus accumbens, right? Going to places like the amygdala for emotional value, right? And those places can produce descending pathways back down to the PDN. So the PDN appears to be, if you look at it neuroanatomically at the connections in the taste system, appears to be a good integrative nucleus to combine signals. We're talking about, well, where does, if I like cheeseburgers today, and that's a positive signal, right? And then I get sick, and tomorrow I no longer like cheeseburgers, where did that change take place? And the PDN looks like an interesting place where all these things converge, right? So it was a target for research to understand how do our taste preferences change with cravings positively or with aversions in a negative sense. All right, and neuroanatomically, if we look at those descending signals back into the PDN, they tend to be, we find a lot of GABA neurons, inhibitory neurons that are projected back there. So from just looking at the rat brain, it looked like, hey, if we want to understand where changes in taste happen, let's look at the PDN and let's look specifically at the role of GABA in the PDN. Okay?
So what does that gray circle represent? The one next to the
This is the nucleus of accumbens. This is our pleasure center, right? Our reward center. All right, so a colleague of mine, J.P. Baird, up at Amherst College, Massachusetts, decided he wanted to try and understand what is the role of the PBN in forming conditioned taste aversion. So the experiment that he did was he gave rats the ability to drink either lithium chloride or sodium chloride. Both of those taste salty to a rat, and a rat can't discriminate between the two. It can't tell from taste if I'm drinking lithium chloride or if I'm drinking sodium chloride. And what he did was measure their licks in 2 4-minute sessions, right? So in the first 4 minutes, the animals haven't been exposed to anything, they're thirsty, they get in there and they start licking, right? And so here they lick a lot to the lithium chloride, right? And lithium chloride makes a rat feel sick. So if we're doing a conditioned taste aversion experiment with rats, the thing that I can inject into their gut to make them feel sick is lithium chloride. Here, I'm not even having to inject it, they're just drinking it, right? So they're actually consuming the thing that'll make them sick. And so they're associating that salty taste with starting to feel sick during that first 4 minutes. Right? Then, so that's here, right? Then in the second 4 minutes, we switch, and now we give them sodium chloride to drink, which won't make them sick, but they're already feeling sick from the lithium chloride, right? So they stop drinking the salty thing. Right? So they lick a lot in the first 4 minutes, 1,500 licks. Second 4 minutes, under 500 licks. They're not licking very much. Right? Now, so that's sort of the model, right? So we can see a conditioned taste aversion form. And in this day, right, for this group, he injected this chemical called anisomycin, right, which is going to interfere with, we think it interferes with memory formations, right? So from learning and memory studies, if you put that in the hippocampus, then you can block long-term memory from happening, right? So he put that in the fourth ventricle, which is where the PBN and the nucleus of the NST are, right? He also put in a vehicle, so a saline. So this is his control group. This is a group that he's saying, hey, if conditioned taste aversions are learned, cannot block the learning by injecting this chemical into the brainstem area, right, where the PBN is. And so then the next day, what he did was he gave them 2 4-minute bouts of sodium chloride, so he didn't make them sick again. But he's testing to see, hey, do they remember the day before? They did form an aversion the day before, do they remember that the next day? And it turns out the group that didn't get, that got the saline, showed very low licking both times, so they showed evidence that they remembered that they got sick to salty things the day before. However, the group that had the anisomycin injected didn't remember that, right? So even though they got sick and showed a taste aversion, the next day they lick a lot more to the sodium chloride, right? So this is pretty good evidence, and one of the things that I love about conditioned taste aversion research is sort of the elegance and simplicity of it, right? It's not a really complicated thing that we're doing, right? But we can learn a lot from a pretty simple behavioral technique. All right, so if we lesion the PBN, right, we don't get taste aversions. If we block activity in the PBN with something like anisomycin, then they don't show a conditioned taste aversion. So this is looking like, what is happening in the PBN, that the PBN may be that site where the change happens, right? And it's remembered, right? And there's a memory trace of that. Right? So the next sort of line of research was looking at some condition preferences. So this is research that came from Tony Sclavani's lab in New York at NYU. And what he did was he's interested in, okay, you have these changes that are happening in the brain, but we also, the sickness is coming from the gut, right? So what post-ingestive signals, how are post-ingestive signals influencing the brainstem? And there are neural pathways from the gut into the PDN. So again, the PDN not only receives descending input from those higher cortical areas, but it also receives input coming in from the gut. So it really looks like this thing that could be modulated by either positive events in the gut or negative events in the gut. And so what he was looking at was positive events in the gut. So he had different flavors of Kool-Aid that were unsweetened, right? So like grape Kool-Aid that doesn't have any calories. And then he was infusing, he could either give them so then he could infuse calories into their gut. So while they're licking the Kool-Aid, he could then inject, if you will, he had a tube implanted where he could infuse sucrose that had calories into the gut. So they never taste the sweetness, they only taste the grape flavor. But one group has, this group here, has it infused at the same time, so 0-minute delay, right? The next group had it 5 minutes after they licked. And then the next group had it 30 minutes after they licked. And so he was able to show that when the calories come into the gut in close time proximity to licking the grape Kool-Aid, at the same time or 5 minutes later, that they show a preference. So the CS negative is water infusion, that's the control. So this is sucrose infusion, this is water infusion, right? So when the calories come in at the same time or very close in time, they show a preference. If we wait 30 minutes and then give them the calories, they don't show a learned association with that grape taste and the calories coming in, right? So there's a window there that the pairing has to happen. So again, using sort of behavioral tasks to understand how do these changes work, right? How do positive increases in consumption due to something like calories or decreases in consumption due to something like getting sick? So 30 minutes was too long to associate taste and calories together. All right, so back to the PDN. So after JP did that study, he and I were talking, and we decided that we would form a collaboration, and we got a grant in order to look at, sort of dissect, if you will, what's going on in the PDN. We said, okay, we have this target, we have some neurotransmitters we think are involved, let's try and dissect this out, right? And try to understand how this happens. And in addition to GABA being an input to the PDN, there's also sort of inhibitory interneurons of GABA within the PDN that we thought, hey, these could be the things that help switch a positive signal to a negative signal, right? This is a mechanism that if we turned on these inhibitory neurons, then maybe that could shape and reroute signals coming out of the PDN. At the same time, there was some research that suggested that benzodiazepines, which is an anti-anxiety drug, like Xanax or Valium, could cause overeating in rats and in people that take these medications. And these are GABA-A agonists. And we're like, hey, if you looked at the experiments that were done with the rats, the overeating, it appeared that they liked the food more, and that's why they were overeating the food. Right? So again, we're like, well, that might make sense if cravings and aversions happen in the PDN, and we're now activating with benzodiazepines GABA in the PDN, then maybe that could be how the changes are, why they're overeating. Right? So our hypothesis was that benzodiazepines do this through increased palatability, meaning makes good food taste better, it makes things that animals would avoid not avoid them as much, right? It makes them taste better. If you make everything taste better, if you shift away from aversions to preferences, then you're going to overconsume that food. Right? And so this is a study that we did, and here we were injecting a benzodiazepine into the whole body of the rat, right? So we're not just focused on certain areas. We will get to where we just put the benzodiazepine in the PBN, but here we're doing whole body things, and if we give them things that they don't like, so if you make salt stronger, they stop licking. Make umami stronger, they stop licking. Bitters, sour, here's another bitter here, right? And what we found is that under the influence of CBP, which is a benzodiazepine, right, the things became less aversive. They licked more, particularly at these aversive concentrations. And that sort of supported our hypothesis that, hey, food that they normally would avoid, they don't want to avoid as much, so they would consume more of that. Right? If we look at things they like sweet, umami, and another sweet here, right? It did the same thing. It increased the palatability, so their consumption went up for things that they normally would like anyway. So in both of these situations, the consumption went up. One question that we had, and one critique of our research was, well, okay, this stuff, you gave them an anti-anxiety drug, maybe they just don't care that this is aversive, right? It's not aversive because it tastes better, it's not aversive because they're under the influence of an anti-anxiety drug. So we gave them capsaicin, which is spicy, but acts through the trigeminal, through the tactile somatosensory system, right? Through the free nerve endings, through the pain sensors, not through the taste system. Doesn't get routed through the PBN or any of those areas. And when we did the spicy, there was no effect, right? So this was sort of our control to say, hey, if they were not avoiding this because they were under the influence of anti-anxiety drug and they just didn't care that it was aversive, they shouldn't care about this either, right? So we showed it was taste specific, right? If we use a non-taste stimulus like a touch stimulus, they didn't show the same pattern. All right, so we published that. We have 4 student co-authors on that paper. And so I always like to point out when we're successful like that. So that was being done in my lab. At the same time, JP in his lab was inserting electrodes into the PBN and measuring the activity of neurons in response to different tastings, right? And so here he was able to go in and measure, hey, there's a group of neurons that respond best to sucrose. Remember we talked about best responding neurons? There's another group that respond best to salt. And then there's a group that responded best to acids, and then a smaller group that responded best to bitter stimuli. And so he would go in and record from a whole bunch of neurons and get these percentages. Then he would inject the benzodiazepine and go back and record again a whole bunch of neurons and look at those percentages. And what he saw was a shift. So after giving a saline injection, these percentages stayed about the same, right? Same percent of sweet best, same percent of salt best, same percent of acid and bitter best. But after benzodiazepines, and this is just whole body injections, he saw a shift in the PBN where now a whole lot more cells are responding to sweet, and a whole lot more cells are responding to salt, two things that rats like, right, or need. And the sour and the bitter percentage shrunk. So here we were able to measure changes in the neural responsiveness of the PBN towards more palatable substances and a decrease in the responsiveness to aversive substances. So now this is getting at, so I'm looking at the behavior, I'm showing this behavior that's happening, and he's showing changes in neural activity that's happening that would support those behavioral results coming out of the PBN. So our next study was, okay, we have a target, we have the PBN. When we give CDP or we give the benzodiazepines in the gut, it's affecting the whole brain, right? What if we now insert a cannula just into the PBN and spritz the CDP just into the PBN? So now we're limiting the effect on all these other areas and trying to prevent benzos from acting anywhere but as GABA agonists here in the PBN. Right? So when we did that with CDP, we saw similar effects. We also used an antagonist of CDP. And when we did that, when we put the antagonist here and we gave the rat a whole body injection of CDP, so the benzos are happening everywhere, but we're blocking their effect only in the PBN, we didn't see the same results. So when we put the CDP in, we saw the same licking patterns that we did with whole body, right? If we do whole body but block the benzos here, we eliminate those effects. So now we've really sort of honed in on the PBN as this really important place that's shifting palatability, and it can be shifted under the influence of these drugs that promote overeating. So this is some of the data from that, showing us blocking the effect with. So here's that effect here with CDP. When we put in the antagonist, it acts just like the controls. Same thing here with CDP, licking goes up to glucose, right? When we block it there, it stays there. So that's where we ended up with that. The last thing that we tried in this series of experiments is that there was a new technique emerging called DREAD. And DREAD stands for designer receptors exclusively activated by designer drugs. Right? And so what that means essentially is, let's see if I can get an explanation of this now. Okay. So what that means is that we could take this modified virus that would infect cells that it is exposed to, and when it infected the cells, it would cause the cells to express a receptor, and that receptor could either be excitatory or inhibitory. And the thing that would open that receptor is a designer drug that has no other effect in the body. So essentially what we do is we take these virus molecules and we inject them. Here we did it into the amygdala, right? That's not lining up. The animation wasn't great. So they actually hit the amygdala and not next to the amygdala. So all the neurons in the amygdala get infected by this virus. And in this case, we're using an inhibitory receptor. And that's going to go down here to the PBN of those axons from the amygdala that are projected to the PBN. So now we can say, hey, we know in lesion studies, you need both the amygdala and the PBN to form a taste aversion, right? So and we've demonstrated that the PBN plays an important role. So what is the role of the amygdala? And so by now having these receptors expressed, when we put this drug called CNO in, which is going to go in the PBN, so we wait 21 days for this virus to be expressed in these axons, right? And then, so now we have DREAD here. We have DREAD's not in the PBN, but only the neurons projecting from the amygdala are there, right? And so then we put in CDP, right, which is going to increase the palatability, increase the licking, and we inject this chemical called CNO that its only thing that it can do is activate those DREAD receptors. So essentially it can silence this pathway from the amygdala into the PBN. So by putting CNO in there, we're shutting down all the activity from the amygdala into here. And when we did that, paired it with the drug, with the CDP, here's an example. So this is the hole, so this is our histology. Here's the hole into the amygdala. And if we take, so this is the amygdala here, this is under a light microscope. We could attach fluorescent molecules to the virus to see if we infected it. And so when we look at it under fluorescence, you can see these are all neurons showing the virus, showing the receptors, right? And we can look at it close, we can see the soma, we can see their projections. And then this is the PBN, and the blue dye represents where the CNO, the thing that would shut down the amygdala pathway, would have gone, right? And so that's where we're saying we're trying to turn off these axons in this area. And again, if we look at it, this is the terminals from the amygdala. So that previous one showed you the actual neurons in the amygdala. This is their axon terminals in the PBN. And here you see all of those receptors that we're turning off in the PBN. You can see over here, outside the PBN, there aren't any axons, right? So it's all limited into the PBN. Yes? Yes, the CNO is the designer drug. Its only job is to turn off all the activity in these red axons here, right? And so when we did this, what we found, if we injected saline or we injected CDP, right, in our controls, and so we also infected the lateral hypothalamus. So we were interested in what is the lateral hypothalamus doing and what is the amygdala over here doing. And what it, and so anyway, our results showed that under controls, we increased licking, right, with CDP. But if we inactivate the lateral hypothalamus, right, then we don't show that increase in licking, right? If we activate so if we use, instead of inhibitory receptors, excitatory receptors, we can actually bump that up a little bit, right? Over here, same thing with the amygdala. When we inactivated it, we shut down the licking, right? So now what we have is our taste perceptions we know can change in positive and negative ways. And the PBN appears to be a place where those taste changes happen, right? The fact that benzodiazepines can induce these changes suggests that GABA is the neurotransmitter that's involved in cravings and aversions in the PBN. And the dread evidence suggests that top-down influences from both the lateral hypothalamus and the amygdala are what are likely activating those GABA neurons in the PBN. So why are the GABA neurons getting activated? Because positive or negative signals are activating the lateral hypothalamus and the amygdala. Wow. Okay, so enough about taste aversions. We're going to look a little bit more at some of the top-down influences. So I mentioned that there are non-taste stimuli in the oral cavity. So we have tactile stimuli. So here we see the fungiform papilla, those big papilla. These little ones, the little dots here in this photograph, are what are called filiform papilla. And these don't have taste buds, but they do have free nerve endings in there, and this is our touch pathway. And so they can detect temperature changes, and they can detect touch as well, mechanoreceptors in there. And they travel in a whole separate pathway. So when I talked about the fact that we didn't see the effect of spicy, because the signal for spicy doesn't come in through a taste nerve, it comes in through the trigeminal nerve, the cranial nerve 5. And it doesn't go to the NST, it doesn't go to the PBN. It does end up in the orbital frontal cortex for flavor perception, but it bypasses all those other areas and goes through our normal touch pathways. So this is doing tactile, so when we talk about the texture of food, right? If mashed potatoes are lumpy or really creamy, smooth, right? That difference in texture is coming in not through taste, but through this tactile system. also temperature. And temperature can play a role in our perception of things in our mouth, but it also plays a role in that retronasal passageway. And so with increases in temperature, those chemicals are more volatile and they're more sort of easily released from our mouth to get up into the olfactory system, right? So we have sort of a lot going on here, right, in terms of in terms of interactions between three systems: taste, touch, and smell, all interacting with each other, right? This is the same system that's activated by spicy things like wasabi and Tabasco sauce that has capsaicin in it. Small amounts can produce heat. Large amounts can produce pain, right? If you've ever eaten just straight wasabi when you're having sushi and you get that tingle in your nose, that's because those chemicals, again, got up and irritated your olfactory mucosa up there and give you that intense sensation. All right. So we mentioned that temperature can affect our perception of taste. And it does so in two different ways. It can change our threshold, our perceptions of salt, sour, sweet, and bitter through actual taste perceptions, interactions on the tongue between the temperature and the transduction mechanisms for taste. Right? It can also, as I mentioned, change flavor, right? So these changes are examples of pure taste, effects on taste, but it also affects flavor. So again, it's sort of hard to dissect those out unless we're doing something like looking at thresholds, not looking at your ability to detect it. And so at higher temperatures, it changes the vaporization of those molecules, and those molecules get easier access to the olfactory, so we enhance the flavor component of that, right? All right. As I mentioned, we have this retronasal passageway, which shows it a little bit better, where, you know, certainly as if you have food that has smell components that you're putting in your mouth, you can breathe that in through the orthodonasal, but more often we're getting this retronasal activation of the olfactory system that's contributing to flavor. And so here we had an experiment that looked at different flavored water. So this is water that's flavored with coffee or wine or vinegar or cherry, all these different flavorings, right? Here's your control, just water. And we asked people to identify, we didn't, but these experimenters did, asked them to identify the flavor. And the black bars is their ability to identify that flavor using their entire flavor system. And then by pinching the nose, you seal off that retronasal passageway. So if I block air from coming in here, it also creates like a dam, if you will, and you can't get air to come in from the retronasal passageway. And when they had their olfaction blocked, their ability to detect and identify, not detect, but identify what flavors those were went way, way down, which tells us that identifying flavors is a key, olfaction plays a key role in that. And this is actually one of the experiments that we'll do today in lab. So you're going to demonstrate your ability to identify flavors with and without your sense of smell. Again, when we damage our olfactory system or it becomes impaired because of something like having been exposed to the coronavirus, or with age, as the olfactory system starts to deteriorate, that influences our perception of flavor and affects our feeding and affects our eating cues. desired. All right, so here's this place, the orbital frontal cortex, right? We have a primary taste pathway, primary smell pathway, we have a primary touch pathway. We also have vision coming in, right? So every single sense except for auditory, hearing, and vestibular combine to produce our sense of flavor. All of those things combine, right? And in addition, the orbital frontal cortex has the ability to project back down and influence things in the lower taste pathway, right? So it's not just an input, but it also has an output that's going back to influence ascending taste signals. Right? So I don't know if you all remember this, but there was a phase when we were probably kids that Heinz decided that they would change the color of ketchup. And the kids would like that, and that would make them want to put ketchup on more things, right? And y'all are looking at it like, no, that wasn't a good idea. And it turns out it really wasn't that great idea. Black ketchup doesn't taste as good as red ketchup, even though it's exactly the same, right? And this is what they thought their perception would be. Once it smiles, I'll put the green ketchup on stuff. But no, it didn't sell very well. People reported that it didn't taste good. And so they didn't buy it. So that fact went away. But this is just a good example of, and we're not going to do that in lab today, but if I gave you a taste test between green ketchup and red ketchup, the only difference is I put food coloring in, it's the same ketchup, right? You would report that the green is less desirable than the red, right? Because of the impact of vision, right? The impact of vision coming into the system. So, again, flavor has more than just the perceptual role. It influences, it interacts with both our hypothalamus, right? And so we talked about if you're really hungry and you go to the pizza buffet and the first slice of pizza tastes really good, after you get full and your hypothalamus starts sending a satiated signal to the orbital frontal cortex, the flavor goes down, it decreases, right? So it both receives input coming in to influence how big the signals are. It also has input coming out to the hypothalamus. So sensory-specific satiety is when, if you overeat, like the example I gave you of the pizza buffet, right, then your perception of that food goes down. So here is, this is with people, and in this study they were given bananas to eat. They had to eat like 10 bananas. As you can imagine, the 10th banana doesn't taste as good. It's probably hard to get it down compared to the first banana that you ate. And then they had them just smell different types of food and rate the pleasantness. And so even just the smell of banana decreased, but none of the other foods showed a change in their pleasantness. So when we become satiated to a particular taste, it only affects that particular food item, that particular taste. Here's the same thing, eating chicken doesn't affect bananas, bananas doesn't affect chicken, vice versa, right? So it's a specific decrease in pleasantness in our flavor perception based on how full we are for that particular food, right? And it doesn't cross over and affect other foods. So this is now doing the same thing, but now we're looking at, with a monkey, looking at the firing rate of the orbital frontal cortex. And here, they are giving them either glucose or black currant juice. And so two things that the monkey likes, right? So starting here, the orbital frontal cortex is firing. These are neurons firing at particular rates, both glucose and the juice. As we now give the monkey more and more glucose, their acceptance of that goes down until they start not wanting to drink the glucose anymore. What's not shown here is the fact that if we give them a sip of black currant, it doesn't change. The pleasantness of that doesn't change. So we're showing a decrease right here, sensory specific satiety. At the same time, look what's happening in the orbital frontal cortex. The neurons are firing less and less and less and less until they get all the way down here where they're barely firing at all compared to spontaneous activity, right? No change in the firing when we give them that sip of black currant juice, right? So a really nice study that's showing, and there was a similar study done with cream, showing acceptance of cream, but a really nice study showing that as the behavioral response goes down, at the same time the firing rate of the orbital frontal cortex is decreasing. So linking perceptions and behavior to changes in neural activity in this flavor area, right? Linking those two together. Right? Same thing here, looking at, this is now, they're satiating them to that black currant juice. So here we see at the beginning, they want to drink a lot, their behavioral response was high. After they consume 100 mils, the behavioral response goes down in each of these. And then they looked again in the orbital frontal cortex, but now instead of giving them the taste, they just gave them the smell of different things. And it turns out that the orbital frontal cortex, when they smell black currant juice, fires a lot. But after they become satiated, the smell doesn't trigger that. Taste, that's the one that we saw in the previous one. Visually, if they see the juice, the orbital frontal cortex fires. But after they become satiated, the vision goes down. So this change in the orbital frontal cortex the change is impacted across all of those sensory systems that are coming in. It's not just the sensory systems. All right, so what does all this mean? Why do we care about this? Well, these types of top-down influences, even more complex top-down influences, such as knowledge and experience, can affect human perception, right? So in this study, people were given 2 samples of wine. It was actually the same wine, poured out of the same bottle. They didn't know that. And in half of the subjects, they were told, they weren't told anything about the wine. They were just asked them to rate the pleasantness of the wine. And they rated the 2 things identical, because they were identical, right? The second group of subjects, they told them that one of the glasses was a $10 bottle of wine, and one of the glasses was a $90 bottle of wine. And they rated the $10 bottle of wine much less pleasant than the $90 bottle of wine, and the only difference was their perception that the wine cost more. Again, exactly the same wine. Right? While they're doing that, they were doing brain scans. They were looking at fMRI data of the orbital frontal cortex. And what they saw here, well, the data is not shown for this, but the response was identical. But if we look at what's going on in the orbital frontal cortex when they drink the $10 wine or the $90 wine, again, the input from the taste, olfaction, vision, touch, all of the sensory input coming into the orbital frontal cortex is identical. The difference is the perception of the cost of the wine. And when they were convinced that it was a cheap bottle of wine, there was a little bit of activation in the orbital frontal cortex and then it shut down. When they were convinced it was the $90 bottle of wine, that activation went way up and stayed up. Right? Again, showing changes in neural activity, right, associated with significant changes in behavior and in perception. Right? So why did they rate this as more pleasant and this less pleasant? Because the activity was actually less in the orbital frontal cortex for the $10 label versus the $90 label. So what's the moral of this? When someone comes over for dinner and they ask about the wine, you tell them it's a $90 bottle of wine. It doesn't matter if it was $3 at all, right? Just tell them it's a $90 bottle of wine. All right, so this pathway is actually, this is a better sort of scientific description of it. Here's the orbital frontal cortex sort of at the center receiving those sensory signals coming in, right? But we also have the lateral hypothalamus and the amygdala interacting with the orbital frontal cortex to influence its activity, right? So when I think it's a $10 bottle of wine, right, the amygdala is shutting down activity in the orbital frontal cortex. When I think it's a $90 bottle of wine, the amygdala is stimulating activity in the orbital frontal cortex. When I'm hungry, right, the lateral hypothalamus is causing the orbital frontal cortex to be more active. When I become full to a particular taste, it's shutting down the activity there, right? So we have motivational states from the hypothalamus, we have reward and perception signals from the amygdala, and again, these are bidirectional, back and forth. So here we see a study looking at activity in the primary taste cortex in response to glucose. So here's the insular, this is the orbital frontal cortex, and here's the anterior cingulate cortex. And so glucose, these are two umami stimuli here that are mixed together. And then this is when everything is presented, all of these are presented together, sort of a composite activity. And what we see is a shift. The glucose is pleasant. People kind of report these umami ones as less pleasant, sort of unpleasant stimuli, particularly at higher concentrations. And we see similar patterns of activation, right, in the primary taste cortex, right? And in the orbital frontal cortex, what changes is in our emotional cortex and the cingulate cortex. When it's glucose, this part of this anterior part is active. When we shift to a negative stimulus, a different part becomes active. Right? And that negative signal is actually stronger than the positive signal because when we put all three of them together, the negative signal outweighs the glucose positive signal, and that area gets activated. Oops. So again, trying to show interactions here between changes in hedonic or emotional areas of our brain influencing our complex perception of flavor. All right. A little bit shorter. In lab, we're going to cover some things that you need to know. I was going to cover them here, but I think this makes more sense. We'll be able to get through it in lab. So we'll talk about the role of genetics and probe sensitivity. We'll talk about correlations between probe sensitivity in your anatomy and changes in coachable papilla and taste sensations. All right. Good. So we'll talk about free fatty acids and fatty taste on Thursday. Get a little extra time today. I'll see you at 2:30 down in the neuroscience lab.